Okay, everybody's back. Okay, thank God. I thought I had lost everyone. Okay, so I am free to take over now at this point. That's excellent. I love being in control. This is fun. Okay, well, let's get rolling. We're back on track here, I hope. So I'm super excited to have an amazing group of speakers here that I have arm twisted a little bit to come and give some of their time and expertise today. So we're going to start off with Jay Forsythe. Jay's in the Department of Chemistry, and I believe they're also biochemistry at College of Charleston. And he will be speaking on analysis of model primordial peptides in complex mixtures. Take it away, Jay.
Will do. Can everyone see my presentation all right? All right. Well, great. Thank you for this opportunity to speak. My name is Jay Forsythe. I'm an assistant professor at College of Charleston, which is in South Carolina in the United States. Like Mike, I teach at a primarily undergraduate institution. So my talk today is going to be a combination of work I did as a postdoc and work that we're doing now with undergraduates. So many of you are probably familiar with the warm little pond hypothesis, right? So Darwin wrote this in a letter to Joseph Hooker in 1871, some years later after On the Origin of Species. And he was kind of thinking about how life could have started, and he proposed this idea of a warm little pond in which different energy sources and chemicals and salts and things like that could have come together to make protein, right? And at the time, he wasn't really sure exactly the role of protein versus nucleic acid, right? Those roles were discovered much later, but this idea has really held up well in the field, right? There was just an article in the BBC just a couple days ago really explaining how his hypothesis is really being explored by a lot of astrobiology researchers. And we essentially do a very similar thing in my lab. So I'm sure that many of you are familiar with the Miller-Urey experiment and analysis of organics on meteorites and things like that. And really the key finding in a lot of those experiments were that amino acids were present in abiotic settings, right? Formed through the Strecker synthesis. But something that may not be as much of a focus is that alpha hydroxy acids or hydroxy acids are also made, right? And so this is a table from a 1959 science paper from Miller and Urey. And you can see that, so glycine, shown here, and its hydroxy acid analog, glycolic acid here, which differ only by the hydroxyl group in the amine, are present in very similar amounts. Likewise, you can see that alanine, the amino acid, and its hydroxy acid analog, lactic acid, are also in very similar amounts, right? So it's generally thought that in prebiotic settings that amino and hydroxy acids were present in similar amounts due to their kind of similar mechanism of formation through the Strecker process. Specifically, I'm interested in how amino acids could have polymerized into peptides and proteins. And in order to do so, they need to condense or form amide bonds and lose water, right? And so this is a reversible reaction. And so if you take two amino acids and combine them, you form a peptide and you lose water. But obviously, water can come back in and attack and send them back to monomers, right? There are a number of ways to carry out amino acid polymerization. This has been studied in the literature for 50, 60 years, right? But what we use is a technique called wet-dry cycling. So you can imagine putting monomers like amino acids in a pool of water, evaporating the water, driving off one of the products and pushing the equilibrium forward and resulting in the formation of polymers. This process does work. However, there's kind of an issue with amino acids that oftentimes they get stuck in a cyclic dimer or a diketopyrazine structure, right? So when you form a dipeptide, this can wrap around, lose water again, and cyclize. And particularly for amino acids, the cyclic dimer is very stable, and it can act as almost a thermodynamic dead end for the reaction. However, when you think about the hydroxy acids, that's not the case, right? And so again, the mechanism is very similar for this reaction. But when you get to this cyclic dimer stage for for cyclic diesters, right, this molecule is much less stable than this, and as a result, you can actually add in, you can attack these ester bonds and lengthen the chain and form these longer polyesters, right? And this has been shown by Mamijanov and others in a 2014 macromolecules paper. So where does my research come in? My research comes in combining these two together. We wanted to know if we could use hydroxy acids and their esters to actually make peptides, right? What happens when we mix these two together? Can we actually grow peptide chains? And the answer to that is yes. These peptides are called depsipeptides. So they're essentially copolymers of hydroxy acids and amino acids, right? So they look very similar to peptides, except they have ester bonds in them as well, right? This is a figure from our 2015 paper in Angomante, and what it shows is that when you polymerize the amino acids and hydroxy acids together, the hydroxy acids react first to form ester bonds, and these activated ester bonds can be attacked by the amino acids to form peptide bonds, right? This reaction is very simple. It's very easy to do. My undergraduates do it in the lab all the time. It takes them a day to learn how to do it. It's very simple. And you can see here, so this is mass spectrometry data. If you do this reaction with glycine alone, you can form short peptides, but not the longer chains don't form. There's really no products above 3, 4 residues in length. However, if you do the hydroxy acid alone, right, you see numerous oligoester polyester products, right? And when you mix the two, you get these combined depsipeptide copolymers, right? An interesting thing about this process is, if you do repeated cycles of wetting and drying, you can actually enrich for the amide or peptide content in the oligomers, right? And we can actually track this by infrared spectroscopy. So if you have an infrared spectroscopy of like a combined, this is lactic acid, which is the hydroxy acid, and glycine, which is the amino acid, after 8 wet-dry cycles, right, you get all these signals. And if you subtract out the signals from the monomers just combined together without any wet-dry cycling, you can actually see what polymerization products are increasing or decreasing with wet-dry cycling, right? And so this is essentially a differential infrared spectrum, right? And so as things go up with wet-dry cycling, we can see them kind of growing and polymerizing. And if things go down, we see them hydrolyzing and breaking down, right? And if you look here, so this is lactic acid and glycine after 1, 4, 8, and 12 wet-dry cycles. And if you look at the amide bonds here, this is an amide 1 vibration, this is amide 2 vibration, and this is also a CN. And you can see that these increase with wet-dry cycling. So it's particularly from 1 to 4 to 8 cycles. The reaction tends to cap out after 8 cycles and under these conditions just due to consumption of the monomer. Whereas the ester bonds, you kind of see them, they kind of reach an equilibrium where they're breaking down and they're building up kind of in similar amounts, right? So with this differential growth of amide bonds or peptide bonds versus the esters, right, if you keep doing this wet-dry cycling, eventually your depsipeptides look a lot like biological peptides. So I'm actually a trained mass spectrometrist, so the majority of work that we do is mass spectrometry analysis. And this is a MALDI-TOF mass spectrometry of glycolic acid and glycine after 8 wet-dry cycles. And you can see all the polymer products here very easily. These oligomers here are 21 units in length, so these are quite long, particularly compared to some other approaches in the field, right? But there's definitely a long ways to go until we get to protein lengths, right? And we have some ideas how to get there, but we're not there yet. And these oligomers here have 8 hydroxy acids and 13 amino acids in them, right? The reason I picked glycolic acid and glycine for this example is if you zoom in, you actually get these sets of peaks. So glycolic acid and glycine are only one mass unit difference, right? And so these peaks on the left have more amino acid, and as they go to the right, they have more hydroxy acid, right? And so all of these peaks here are actually clusters of peaks, which we can very easily tell apart. And this work was published in 2018 in Rapid Communications and Mass Spectrometry, and my student Sloan English was an undergraduate, and she performed all this work herself. So the last thing I want to talk about, I don't have much time, is a paper we published in PNAS in 2017 with more complex combinations of hydroxy acids and amino acids, right? So we have glycine, alanine, leucine, and then hydroxy as we have glycolic acid, lactic acid, and malic acid. And so we did a combination of all of these after 1, 2, and 4 cycles. We did liquid chromatography, ion mobility spectrometry, which is gas phase electrophoresis, and then sequenced all the peptides by tandem mass spectrometry. And the mixtures were extremely complex, right? We use these kind of multi-dimensional separations to really pull everything apart. These bubbles represent individual DEPC peptide combinations, and the color, like how the darker the red, the more amino acid, and the lighter the red, the more hydroxy acid. And we also did, we used web logos, which are used to monitor like sequence homology and sequence diversity. And if you look at these examples here, maybe just for time, I'll just focus on this one example here. So these are glycolic acids here on the first residue, and then the second, third, or fourth residue could have glycolic acid, alanine, glycine, or leucine. And what you can see here is these letters shrink with cycling, and what that means is that the sequence diversity of these positions is increasing with wet-dry cycling, and it's not the same for all amino acids, right? So you can see here that there's a lack of leucine after 1 cycle, but after 4 cycles we have the letters shrink, which means we have more leucine, right? And so we can actually track the sequence diversity by mass spectrometry as well, which is really exciting looking forward. So I will go ahead and stop there. I think I'm running out of time. This is a picture of my collaborators from the Center for Chemical Evolution, and this is a picture of some of my undergraduates from College of Charleston. And with that, I will go ahead and stop and take questions. Thanks.
Good stuff. Thanks, Jay. Very interesting work. Awesome to see you've got undergraduates really dialed into some of this very, very cool work. I'm not seeing any questions in the chat room. Jump those in, or you can, let's see, there's only a handful of us in here, so I suppose you could unmute and ask that question. I am curious, Jay, in that plot you showed where I think the smallest peak you showed was the 8G, small g plus 13G. Mm-hmm. Each of those peaks is a concentration, is reflective of the, yeah, right there. So when you get down to, what is that, 1200 plus, have you reached the limit of the synthetic ability there? Is that what we're getting out of that plot?
That's a really good question, right? So you can almost see, like, it's almost kind of like a statistical curve. Yep. It goes down as you get longer lengths, right? And so that's fairly common with what we see. You know, I think it's kind of like a random, like at least with this combination of monomers, it's kind of a random polymerization, right? So statistically, sometimes you see that. It's also so it's important to recognize that the intensity of mass spectrometry peaks are not only due to their abundance, they're also due to how they ionize, right? Right. And so sometimes some of these shorter ones actually have, they ionize more easily than some of the larger ones. It depends on a number of factors, but I think in general, we actually we tend to see kind of them cap out around 20 residues in length. And okay. So I think what we're going to need to do is start throwing in other like catalytic materials, kind of like we were talking about with some of your minerals or things like that, maybe to start get us past that hump. It also depends on the how hydrophilic or hydrophobic the residues are, right? If you have a lot of hydrophobic amino acids or things like that, sometimes they're shorter because they crash out, right? Yeah. And so I'm really interested in getting super complex mixtures of polar and non-polar amino acids because one, I think that'd be more plausible prebiotically, but two, I think that actually might make them longer because you'll have more residues that will keep them in solution, right? Yeah, yeah. Yeah, I was definitely struck by that sort of quasi-exponential drop there. And having less experience with that type of mass spectrometry, I was just curious how that was interpreted. So, all right, very cool. Questions from anyone else in the session here? Okay, great job, Jay. Thanks a bunch. Great job staying on time. Definitely appreciate that. For those of you just joining in, we were about 15 minutes going late. So let's be as diligent as we can.